Hello, this is Robert Murphy again, the University of Colorado. And this short video is going to be concerning a unique tool that is available from Avanti Polar Lipids that can facilitate studies of aminophospholipid and measure very subtle changes in their concentrations in various biochemical studies. In this video, we're going to talk about a technique where one can measure subtle changes in aminophospholipid levels using a derivatization step with dimethylaminobenzoic acid stabilized-isotope isomers. And it's an interesting method that allows you to multiplex experiments. And so I'll try and explain what all of that means with a few slides. So this is now the phosphatidylethanolamin species, which are present in mouse heart, and you see there are quite a few species, some major, some minor, and this once again is an experiment where it's from a knockout animal versus a control, the red being the knockout animal, control being the blue, and you can see that there are changes in the red and blue traces and whatever these species are. So let's say you have an experiment where you think that you may have a drug which is changing the Lanz pathway of phosphatidylethanolamin reacylation, and you want to look at the effect of the drug at different doses, and you want to be pretty accurate in what changes there are. And let's say that you're looking at as many species as you can, but you want to see how these levels are changing, and you're not willing to accept that you're going to be 10% accurate or 15% accurate. You really want to be pretty precise. So the problem with that is, of course, is you can't add, as I've mentioned before, internal standards for each and every one of these molecular species of phosphatidylethanolamin because, well, first of all, they're not available. But even if they were, there's really very little space for you to put very many of them. There's a few empty spaces, but not many. And in fact, every spacer either has a phosphatidylethanolamin molecular ion or it's C13 isotopes present. So how do you deal with this? What can you do? Is just you can't do the experiment? No, there's a way we can get around this. And so this one takes advantage of the power of stabilized-isotope dilution strategies and the value of these internal standards for each and every one of these molecular species. So example here, we would follow with a drug. We could maybe see what effect diets have on these two knockout animals and so on like that. But we want to measure subtle differences between individual species of this class of lipids, these aminophospholipids. So this is a challenge, and to measure these subtle changes for each one of these, either as the positive protonated molecular ion or as the negative ion, we really have to come to grips with the fact that each one of these has a very different ionization efficiency and stability. And those are the major driving factors which are going to change that relationship between ion abundance and ion concentration, or PE concentration. So the solution to this is what I propose to you is this novel isotope-labeled dimethylaminobenzoic acid derivative standard. So these are the reagents which are available from Avanti. They're N-hydroxysuccimide esters of dimethylaminobenzoic acid. So I'll point out this structure here. Here's dimethylaminobenzoic acid, and here is the activated carboxylic acid as the N-hydroxysuccimide ester. They come in 4 different flavors. A non-deuterated form. Here's one that's got 4 deuteriums on the aromatic ring. This flavor has 6 deuteriums on the 2 methyl groups of the amino groups on the nitrogen, and then this version has both, 4 on the ring and 6 on the dimethylamino group. So there's D10. So we have D0, D4, D6, and D10. So when we make these derivatives of the PEs, they are going to have exactly the same properties in terms of ion efficiency and stability, but they will have different masses we can measure in the mass spectrometer. So this is the chemistry which is carried out. We take these NHS esters, we incubate them at elevated temperature, and then we add water to hydrolyze the excess reagent, and then we do blydier extract. And so the phosphatidylethanolamine indicated by this structure here, the lone pair of electrons, of course, attacking the ester derivative here, and this succinimide is a very good leaving group, N-hydroxysuccinimide is a good leaving group. And so we now have these amides, which are products with PE, and so they'll be either labeled with the D10, the D10, the D4, the D6, or non-deuterated. So the interesting feature about these molecules is their tandem mass spectrum. So let me point out what these features are. So this is the positive ion tandem mass spectrum of these derivatives. So they have a nice molecular ion, you collisionally activate it, and it forms really two really abundant ions. The one ion, 191 in this case, comes from this group, which I'll now call the reporter head group here. And this is either going to be 191 for D0, 195 for D4, 197 for D6, or 201 for D10. So it really tells you which one of the labels you have. The other major ion is the lipid portion of the molecule. Very common feature of positive ions of all phospholipids is you have these diglyceride-like ions, and it's constant, 603, 603, 603, 603, because we derivatize the same lipid, the di-16PC, 18-1 PE. We derivatize those, and so they have the same one. If we have a different PE, it'll have a different diglyceride mass. The negative ion tandem mass spectrum of this molecule, they also make very good negative ions, really reveals the carboxylate anions. So that's the same cleavage reaction we talked about in the first lecture, and you see these carboxylate anions become abundant product ions. In this case, it's the same carboxylate anion, so we only see 281, 281, 281. And of course, the molecular weight tells you how many deuteriums there are in that, but this allows you to reveal what are the carboxylate anions. So we're going to use that to try and understand something about some events which are taking with a mixture of phosphatidylethanolamines. So this is the experiment. It's a very contrived, straightforward experiment. We took some experiments, I would call it multiplex it, and use this differential display to really try and find out what's going on. So we take some cells, we extract the lipids with a chloroform methanol extraction protocol. We make liposomes out of these lipids, and now we add to these liposomes a free radical generating system, AAPH, which under constant condition, temperature, and time forms free radicals. We add to this mixture a reporter internal standard, and you'll see the importance of this later, PE 14-0, 14-0. And then at various time points, 0, 30, 60, 300 minutes, we're going to take aliquots out of this mixture, and then we're going to subject them to the derivatization strategy. The 0 minute will add D4 DMABA. At 30 minutes, we'll add D4. Sorry, that was D0. This is D4. 60 minutes, we'll add D6. 300 minutes, we'll add the D10 DMABA. And so each one of these time points is now going to be marked by the deuterium-labeled reporter. Now we take all of those derivatized samples and we mix them together. Make one sample out of it. And so now all of them are mixed together, and now we perform a single LCMS run, and either run a positive ion as one run, a negative ion another run, or with newer machines we can run positive and negative in the same experiment. And actually in this experiment, I misspoke. We have these labeled a little bit different. The control is the 191, that's D0. This is D4, 30 minutes. 60 minutes is the D10, and D6 is 300 minutes. So that's the color code you're going to see. So now we'll look at what the molecular species look like. And this is sort of now a trace of the experiment in which we're going to be looking at product ions, 191, blue trace. You can sort of see a little bit in here. Most of it's coming out here. The green trace, which is 30 minutes, which we're getting some peaks in this early region, but also here in this later region, a lot more of those. 201, a little bit less in this later region, and 197, quite a bit less in this later region, but now quite a bit more in this early region. So what are all these things? These are telling us, when we do these precursor ion scanning, they're telling us which ones were coming off depending upon the time of incubation with that free radical generating agent. So we see this red one here. These are the early peaks, and so they're being formed much later. So they're probably the products of more extensive free radical oxidation. The other feature I want to point out is this internal standard. You see, I've normalized everything to the same internal standard. So this molecule is not affected by that free radical process. And you can see that its height has been normalized. So we knew we took the exact amount of sample from each one of those aliquots so that we're looking at equivalent amounts of lipid originally from the raw cells. So now if we take a look at the traces and we look at positive ions, 633 in the red trace, the precursor 197, that's the D6. We see we have ions at 633, 655, 689. There's a lot more of these ions in here. And the blue one is the corresponding same molecule, but now has D0, and there's precious little, and certainly none of this one as 633. So where does this 633 come off? Well, we extract 633 from the data we've generated, and we see, well, it's a single component coming at 10.2 minutes. And that's what comes off. So what is that ion? We know its mass is 633. We know it's a derivative, but we don't know what molecular species it is. So now we go in and look at the negative ions of now 631, 2 mass units different because we're looking at negative ions. And now we see it's got 281, there's a big peak here, and not much else. And if we add the mass of the PE and the derivative and add now 281, we are left with 631. So this has to be a lyso PE product. So a free radical product of oxidation of these PE species are lyso PE species. If we look at another ion, which is quite abundant a little bit later, it was the one at 19.0 minutes, we see much more complex number of red peaks, and they're virtually all being derived from this oxidation experiment. In other words, the blue trace, which is time 0, there's none of this. But the chromatographic peaks are really complex. They're all adding together, and they're not forming a nice single peak. So when we look at the negative ions of this peak, we see that really the mass of this ion, which is 953, and the negative ions 951, we can look at these product ions, and we don't see just a characteristic acyl group. What we see these ions, which are very characteristic of prostaglandins. And in fact, these things probably have these structures here. This is the 167 portion of the molecule, which we now can assign as to be an ether phosphatidylethanolamin. But the SN2 position is a cyclized arachidonate in a process which we call isoprostanes. So these are families of molecules. In fact, this one structure here is a family of 64 different structures, all of which have different retention times, and so this causes this smear right here. And this is a well-known process of free radical oxidation forming isoprostanes. Now, one of the things we do to verify that is to actually derivatize that ketone group by a methoxylamine derivative, and we do that in the gas phase, and I'll just show you what that is. We put the sample in a capillary tube, and we seal it inside there, pull a vacuum, and then we heat up a solution of methoxylamine hydrochloride with some sodium hydroxide, and then methoxylamine is now a gas, and it goes into the sample and derivatizes it. Now, the last example of one of the new peaks was this later peak, which was coming off. Once again, here's the red peak. There's lots of ions in this region, around 15 minutes, lots of ions. There are lots of different products coming off. And that's the story of free radical oxidation. It makes lots of products. So you sequentially go and investigate each one of these. We'll just do one of them. We'll do the one peak at 7:46. You see it has a nice chromatographic peak at 15.7 minutes and then another one at 14 minutes. And when you switch to the negative ion data and you look at that, you see there's a big peak for 144, and that's about it. So that is another plasmalogen PE, and here is the chain shortened 5-aldehyde, which is now a methoxine because we derivatized this, so that's 144. So you can identify that as a chain shortened phospholipid by that free radical process. So all this data is obtained in a single LC-MS run. If you can do positive negative ion switching, you go in, and this is just data, just searching through the data, the mass spec data, finding the information to allow you to draw the structures. And you're able to do that because you have these tags on here which allow you to pick out the unique structures which are products of time-dependent free radical reactions, because we're looking at things which are not normally present. They're not normally blue. Those later peaks, which I didn't show you, those are all naturally occurring PEs, and they were being lost in abundance, but they were just the hundreds of different PE molecular species which were being oxidized. So here's some references to using this strategy. It's sort of a very esoteric strategy, but one which is available from Avanti, those deuterium-labeled reagents. And if you're interested in phosphatidylethanolamine or phosphatidylserine biochemistry, they can be tools which allow you to investigate unique biochemical events taking place with these aminophospholipids.